ABCA4, Wikipedia article audio. NM000350. NM007378. Structure. Function. NP000341. NP031404. ADP binding cassette, subfamily A, member 4, also known as ABCA4 or ABCR, is a protein which in humans is encoded by the ABCA4 gene. ABCA4 is a member of the ADP binding cassette transporter gene subfamily A found exclusively in multicellular eukaryotes. The gene was first cloned and characterized in 1997 as a gene that causes Stargardt disease, an autosomal recessive disease that causes macular degeneration. The ABCA4 gene transcribes a large retina-specific protein with 2 transmembrane domains, 2 glycosylated extracellular domains, and 2 nucleotide binding domains. The ABCA4 protein is almost exclusively expressed in retina localizing in outer segment disc edges of rod photoreceptors. Previously known as the photoreceptor rim protein RMP or ABCR, the recently proposed ABCA4 structure consists of 2 transmembrane domains, 2 large glycosylated extracytosolic domains, and 2 internal nucleotide binding domains. 1 TMD spans across membranes with 6 units of protein linked together to form a domain. The TMDs are usually not conserved across genomes due to its specificity and diversity in function as channels or ligand binding controllers. However, NBDs are highly conserved across different genomes, an observation consistent with which it binds and hydrolyzes ADP. NBD binds adenosine triphosphate molecules to utilize the high-energy inorganic phosphate to carry out change in conformation of the ABC transporter. Transcribed ABCA4 forms into a heterodimer. The 2 dimerized compartments of the channel are different from each other. When TMDs are situated in a membrane, they form a barrel-like structure permeable to retinoid ligands and control channel access to its binding sites. Once an ADP is hydrolyzed at the NBDs of the channel, NBDs are brought together to tilt and modify TMDs to modulate ligand binding to the channel. A recently proposed model of retinoid transfer occurring as a result of alternating exposure of external and internal TMD ligand binding sites, all controlled by binding of ADP, is based on recent structural analyses of bacterial ABC transporters. Clinical significance. ABCR is localized to outer segment disc edges of rods and cones. ABCR is expressed much less than rhodopsin, approximately at 1:120. Comparisons between mammalian ABCA4 to other ABCs, cellular localization of ABCA4, and analyses of ABCA4 knockout mice suggest that ABCA4 may function as an inward-directed retinoid flip passe. Flip passe is a transmembrane protein that flips its conformation to transport materials across a membrane. In the case of ABCA4, the flip passe facilitates transfer of N-retinal phosphatidylethanelamine, a covalent adduct of all trans-retinaldehyde with phosphatidylethanelamine, trapped inside the disc as charged species out to the cytoplasmic surface. Once transported, ATR is reduced to vitamin A and then transferred to retinal pigment epithelium to be recycled into 11-cis-retinal. This alternating access release model for ABCA4 has 4 steps: binding of ADP to an NBD to bring 2 NBDs together and expose outer vestibule high affinity binding site located in TMD, binding of NRPE/ATR on extracellular side of the channel, ADP hydrolysis promoting gate opening and movement of NRPE/ATR across the membrane to the low affinity binding site on the intracellular portion of TMD, and release of adenosine diphosphate and inorganic phosphate to release the bound ligand. The channel is then ready to transfer another molecule of NRPE/ATR again. The ABCR/knockout mouse has delayed dark adaptation but normal final rod threshold relative to controls. This suggests bulk transmembrane diffusion pathways that remove ATR/NRPE from extracellular membranes. After bleaching the retina with strong light, ATR/NRPE accumulates significantly in outer segments. This accumulation leads to formation of toxic cationic bis-pyridinium salt, N-retinylidene N-retinyl ethanolamine, which causes human dry and wet age-related macular degeneration. From this experiment, it was concluded that ABCR has a significant role in clearing accumulation of ATR/NRPE to prevent formation of A2E in extracellular photoreceptor surfaces during bleach recovery. Mutations in ABCA4 gene are known to cause the autosomal recessive disease Stargardt macular dystrophy, which is a hereditary juvenile macular degeneration disease causing progressive loss of photoreceptor cells. STGD is characterized by reduced visual acuity and color vision, loss of central vision, delayed dark adaptation, and accumulation of autofluorescent RPE lipofuscin. Removal of NRPE/ATR appears to be significant in normal bleach recovery and to mitigate persistent opsin signaling that causes photoreceptors to degenerate. ABCA4 also mitigates long-term effects of accumulation of ATR that results in irreversible ATR binding to a second molecule of ATR and NRPE to form dihydro-N-retinylidene N-retinyl phosphatidylethanolamin. A2PEH2 traps ATR and accumulates in outer segments to further oxidize into N-retinylidene N-retinyl phosphatidylethanolamin. After diurnal disc shedding and phagocytosis of outer segment by RPE cells, A2PE is hydrolyzed inside the RPE phagolysosome to form A2E. Accumulation of A2E causes toxicity at the primary RPE level and secondary photoreceptor destruction in macular degenerations. Additional diseases that may link to mutations in ABCA4 include fundus flavomaculatus, cone rod dystrophy, retinitis pigmentosa, and age-related macular degeneration. The Geneva Cleft Consortium study first identified ABCA4 as being associated with cleft lip and/or cleft palate with multiple markers giving evidence of linkage and association at the genome-wide significance level. Although SNPs in this gene are associated with cleft lip/palate, there is no functional or expression data to support it as the causal gene, which may, instead, lie in a region adjacent to ABCA4. A combination of genome-wide association, rare coding sequence variants, craniofacial specific expression, and interactions with IRF6 support a role for the adjacent RHGAP29 gene to be the likely causal gene playing a role in non-syndromic cleft lip and/or palate. This article incorporates text from the United States National Library of Medicine, which is in the public domain.